All right, today we're going to unpack the story of the Mpox virus. In just the last few years, this one pathogen has managed to trigger two separate global health emergencies, and it's really forced us to re-examine everything we thought we knew about it. So let's just dive right into the virology and see how we got here.
And here it is. This is our subject. Under the electron microscope, the monkeypox virus, or MPXV, it's got that classic, almost brick-like structure that you'd expect from any orthopox virus. It's a really complex piece of machinery, and as we're about to see, its comeback is posing some pretty fascinating and, frankly, urgent challenges for global health.
So that really brings us to the core question of this whole case study. We're not just here to go through a timeline of events. What we're really asking is, what can this specific virus teach us about modern virology? And maybe even more important, what does it tell us about the vulnerabilities in our global health security? To really get to the bottom of this, we're going to break our analysis down into 4 parts. First, we'll look at the anatomy of the virus itself. Then, we'll trace its journey from being a pretty localized endemic problem to a global one. Third, we'll dig into the 2 very distinct emergencies, which were driven by different viral clades. And finally, we'll take a look at the public health toolkit we actually have to fight back.
Okay, let's get into Section 1, the agent. Before we can even begin to understand the disease, we've got to understand the fundamental virology of the monkeypox virus.
So its official classification places it in some pretty notorious company, right alongside variola, the virus that causes smallpox. As an enveloped, double-stranded DNA virus, it has a large and what we'd consider a relatively stable genome, especially compared to a lot of RNA viruses. But, of course, stable doesn't mean it can't evolve, as we are definitely about to see.
Okay, now this is where the molecular details get really interesting. So in Figure A, you can see that complex structure. You've got the DNA core, the lateral bodies, all of it wrapped up in multiple membranes. But for our purposes, Figure B is absolutely crucial. The genome is massive. We're talking around 197 kilobase pairs. Now, the central region is conserved. It holds all those essential housekeeping genes for replication. But the real action, the interesting stuff, is happening in the variable terminal regions. That's where you find the inverted terminal repeats, or ITRs. These are critical for kicking off replication, and they're just packed with genes that help the virus dodge the host's immune system.
Okay, so let's move to our next section and see how all this virology plays out in the bigger picture. We're going to trace the journey of MPXV from what was a scientific curiosity to a global headline.
This timeline really illustrates the virus's slow burn. It was first spotted way back in 1958 in lab monkeys in Denmark, of all places. Then it took another 12 years before the first human case was even confirmed. For decades, it just kind of simmered, causing sporadic cases, often getting mistaken for other diseases. But then you see this acceleration. The jump to the US in 2003, a really significant outbreak in Nigeria in 2017, and then boom, the explosion onto the global stage in 2022.
And this is that critical moment, that first documented jump from animal to human, confirmed in 1970 in the Democratic Republic of Congo. This really established Central Africa as the crucible for this virus's relationship with our species.
Now, this next point is such a critical piece of the puzzle, and a fantastic example of the unintended consequences of a massive public health victory. When we eradicated smallpox in 1980, the world stopped routine vaccination. And while that was a monumental achievement, it also meant that the cross-protective immunity that vaccine gave us against other orthopox viruses, including Mpox, well, it started to fade away. Whole generations grew up without it, creating this vast immunologically naive population, which is just the perfect environment for a forgotten virus to make a comeback.
And it is absolutely essential to understand that this virus isn't monolithic. It's not just one thing. As this map shows so clearly, there are 2 distinct lineages, or clades. You have Clade I, which is endemic to the Congo Basin in Central Africa, and then you have Clade II over in West Africa. This geographical and genetic separation is fundamental to understanding the 2 emergencies we're about to talk about.
And that brings us right into the modern era, and really the core of our analysis today. These 2 distinct global health emergencies, each one driven by a different clade of the very same virus.
The contrast here is just so stark. On the left, you've got July 2022. The World Health Organization declares a public health emergency of international concern, a PHEIC, for a global outbreak. Then on the right, just 2 years later, August 2024, another PHEIC is declared, but this time for an escalating outbreak centered in Africa. Same virus, 2 emergencies. So what's going on?
Well, the answer lies in the clades. The 2022 global outbreak was all driven by Clade II, that's the West African lineage, which historically has been associated with less severe disease. But the more recent 2024 emergency, that's being driven by Clade I, the Central African strain, which has a history of causing much more severe clinical outcomes. This distinction isn't just academic, it is everything.
So, what do we actually do about it? In our final section, we're going to open up the public health toolkit and analyze the countermeasures, the scientific and medical tools we have at our disposal to diagnose, treat, and hopefully prevent Mpox infection.
You know, understanding transmission is always the first step to prevention. In nature, the virus just hangs out in a zoonotic cycle, likely involving rodents and primates. It spills over to humans through direct contact or even through the consumption of bushmeat. And from there, human-to-human spread happens through close physical contact with the infectious rash, scabs, or body fluids. Now for treatment, let's zoom in on the viral life cycle to find some potential points for intervention. This is a great diagram. You can trace the entire process from the virus getting in to its DNA replication inside those cytoplasmic factories and finally assembly and exit. When we're looking for antivirals, we're looking for a chink in this armor. So take a look at step 8, exocytosis. The drug tecovirimat actually targets a protein that's essential for that final wrapping of the virus before it leaves the cell. It basically traps the new virions inside, stopping them from getting out and spreading.
And building on that, our current antiviral arsenal pretty much relies on 3 main drugs, and they were all repurposed from our fight against smallpox. So you've got tecovirimat, which you just talked about, along with cidofovir and brincidofovir. They're often used to manage severe cases. But it is so crucial to remember that we are still in need of purpose-built proven therapies that are designed specifically for Mpox.
And this quote from Hugh Adler, which came from the height of the 2022 Clade II outbreak, it's just a really sobering reminder. It shows that even with our advanced molecular understanding, and with vaccines and antivirals, containment is incredibly difficult. Public health strategies really struggle to keep pace, which just highlights how hard it is to translate what we know in the lab into effective success out in the field.
We're going to end on this question, because the story of Mpox isn't really just about one virus. It's a powerful case study, and it's a very clear signal. As human activity keeps pushing further and further into animal habitats, the opportunities for these zoonotic spillover events are only going to increase. The reemergence of Mpox is both a lesson and a warning. So the real question for all of us is, what does this mean for our future? And what's our response going to be next time?